Okay, thank you for being here. I'm Amelie. I'll be presenting some of the work from my lab group at the University of Virginia, led by Peter Berg and Karen McGlathery. And I'll be presenting our research on seagrass metabolism and carbon burial on Virginia's eastern shore. And this is kind of a hot topic nowadays because seagrass ecosystems are highly productive and they actually retain about as much carbon as terrestrial forests do, even though they only cover about 0.1% of the sea floor. So they are very important components to the global carbon cycle, and they're often overlooked. So to start, our study site is the Virginia Coast Reserve, which is on the Atlantic side of the Delmarva Peninsula. There's a Chesapeake Bay. And the system consists of these shallow coastal lagoons that are located between barrier islands and marshes. These lagoons once supported extensive seagrass meadows, but in the early 1930s, we had this eelgrass wasting disease come through, which decimated eelgrass populations across the entire North Atlantic. Add on top of that a storm that hit the region around the same time, and we lost all our eelgrass populations in the area. Now, unlike other bays that are to the north and south of this area, we didn't see a recovery here, mainly because we lost our seed supply. So it wasn't until the 1990s that a natural patch of eelgrass was found in these lagoons, and this spurred this whole large-scale restoration program that started in 2001 and was led by Bob Orth and his group. So you can see pictures of them here collecting eelgrass seeds and literally just throwing seeds into the lagoons to restore the seagrass habitat. So since 2001, 500 acres have been seeded, and less than 2 decades later, we have about 25 square kilometers of restored seagrass habitat now. Most of our research is focused in South Bay, which is kind of recognized to this day as the largest successfully restored eelgrass meadow. It's also easier to access from our lab. So this is just a map showing you the seagrass extent in South Bay as the meadow grew from its original seeded plots to its present extent of over 7 square kilometers. We've been following the development of this meadow by doing annual surveys during peak growing season, usually in the summer, and where we go out and we count seagrass shoot density, we sample seagrass tissue and sediment for their carbon and nitrogen content and so on, so we have a long-term record of that. And then in 2007, we started doing seasonal measurements of seagrass metabolism using the aquatic eddy covariance technique. So if you look at seagrass shoot density versus meadow age, you can see a clear period of ecosystem development until about 9 years out, where the meadow reached this relatively stable, mature state. But in 2015, we had a very high temperature event that led to this large-scale die-off in the meadow. And as you can see from this picture, it's pretty dramatic. This is also what it looked like when I started my PhD, and I was like, that's not what I thought it would look like, because it all died. But that gave me a project, so that was good. So in the years following this die-off event, we have been observing a slow and partial recovery. The recovery does depend on where you are in the meadow, and I'll get into that a little bit later. But our annual surveys and our aquatic eddy covariance campaign kind of captured this event. So this allows us to look at the carbon retention capacity of this seagrass meadow and how that is affected by disturbance events such as a marine heat wave coming in. And that's especially relevant as we're about to get more and more in decades to come. So I'm going to start by talking about this long-term record and kind of establishing the baseline of seagrass metabolism in the meadow, and then I'll go into the details of the die-off event and the original restoration as well. got a shout out to Aquatic eddy covariance. My lab is known for using this technique in a variety of benthic environments, including oyster reefs, coral reefs, riverbeds, undersea ice, and so on. But our longest running measurements have been done in South Bay. So we have this really nice long-term record that no one else has. It's pretty cool. The aquatic eddy covariance technique does provide this very nice data. So this is an example of a 3-day deployment in South Bay. And you can see this really nice coupling between light levels and the oxygen benthic exchange. And when you have a lot of this type of data over multiple days, you can start to extract more detailed information about benthic metabolism. This is just very quick to show you how we get to our metabolic numbers. We separate our data into 24-hour segments, and then we use these hourly fluxes in these equations to calculate respiration, gross primary production, and net ecosystem metabolism, which is the difference between the two. And so now we have about 115 daily values for these metabolic parameters that span a period of about 11 years. And what jumps out when you look at all that data is how dynamic seagrass metabolism is on various time scales. So we're looking at the daily time scale here. These are daily values of respiration versus gross primary production, plotted relative to this 1-to-1 line, which indicates metabolic balance. So if the points fall on this side of the line, the meadow is heterotrophic, so it's a source of carbon. If points fall on this side of the line, the meadow is autotrophic, just taking up carbon. And just to clarify, this is benthic metabolism as a whole, so it includes the seagrass, the algae, critters, it's just the whole ecosystem. So we do see a lot of day-to-day variability between heterotrophy and autotrophy, but overall the points do fall close to this line, so that indicates that on average the system is in metabolic balance. We can see this on a monthly time scale as well. So now what you're looking at is our long-term record plotted as monthly averages of gross primary production and respiration. And then in the middle here we have net ecosystem metabolism and seagrass shoot density at the bottom. You can see the die-off, by the way, and the partial recovery. We're still on an upward trend, good news. But you do see here again that it's pretty dynamic, but respiration and gross primary production do mirror each other pretty well, which results in relatively low net ecosystem metabolism values that are generally not different from 0, except here where for the months that are marked by asterisks. And if I haven't convinced you enough that our system is in metabolic balance, we reconstructed an average annual oxygen budget for the meadow, basically by binning all our data by month of year. So this kind of integrates over this 11-year record that we have and shows you respiration and gross primary production for each month. And so then we get these annual values. This is in millimoles of oxygen per meter squared per day. And we get a very insignificant net ecosystem metabolism of 1. This is also a very convenient result because it allows us to kind of map out an annual carbon cycle for the meadow. Unit change alert, we are now talking in moles of carbon per meter squared per year. And so what our results have allowed us to do is, since respiration and gross primary production are basically equal, that must mean that the net inputs of carbon to the meadow are matched by burial and suggests a fast internal cycling of carbon within the system. All right, so now that we've established that on average over this entire record, the seagrass meadow has been in metabolic balance, we're going to look at carbon sequestration and storage on an interannual time scale. So this is where the restoration and die-off event come in. So restoration reinstates carbon storage in the sediments. So this is based on our annual surveys. We looked at how much carbon was stored in the top 5 centimeters of the sediments and found that the restored sediments after about 12 years stored about 1.6 times more carbon as unrestored sediments in this brown bar. Most of that sediment carbon is from seagrass tissue. About half, the other half is from in situ algal production, so it makes sense that an expanding meadow would contribute to increased carbon storage in the sediments. The remaining 10% of that sediment carbon is from outside sources and gets trapped in the meadow by the canopy or settles out of the water column due to reduced flow conditions. And now moving on to carbon sequestration, which is just the uptake of carbon through photosynthesis. Naturally, we found that restoration increased carbon sequestration. So you can see here the rates of respiration and primary production increasing 10 to 13 years after restoration relative to unrestored sediment. So restoration increased both carbon sequestration and storage in the sediment. Now, what happened with this die-off event? So in the summers before the die-off event, the meadow was nice and mature and lush and metabolically balanced. But when the die-off event happened, again, we saw a dramatic decrease in shoot densities that was accompanied by a dramatic decrease in ecosystem respiration and gross primary production. The trophic status of the meadow shifted from being balanced before the die-off to being heterotrophic right after the die-off. It kind of makes sense. We lost most of our photosynthesizing biomass, and we think that microbial respiration was also stimulated because of all this dead seagrass tissue that was now being decomposed. Now, during the recovery, we saw an increase in seagrass shoot density along with an increase in respiration and gross primary production. The meadow shifted again towards net autotrophy, so being a sink of carbon. Mostly that is the result of all this new seagrass biomass that was growing in. So now moving on to what happened in terms of carbon storage. This graph should look familiar. I showed you the left part earlier. So this is, again, all the carbon that was stored as a result of the restoration project. We lost it all within 2 years of the die-off event. So that's the bad news. The worst news is that most of that carbon tends to get remineralized, and so you get emissions of CO2 from carbon that had been buried over decades to, in some cases, millennia. So that's not good news. But we are now storing carbon again as the meadow has been recovering, even though there is somewhat of a lag behind the recovery in seagrass shoot density. A little bit more good news is we did not lose all that carbon everywhere in the meadow. It very much depended on where you were. So this is an aerial photograph from 2 years after the die-off. And I don't know if you can see it that well on the PowerPoint, but you kind of see this lighter color in the center here is where the seagrass is still very sparse, recovering much more slowly. But you see a lot of seagrass around that, particularly near the inlet. And so this pattern in seagrass shoot density does match what we found for seagrass sediment carbon. So this graph on the left shows you for the near inlet site and the mid-meadow the change in carbon storage from before the die-off in blue and then after the die-off in green. So you can see that near the inlet, we didn't lose all that carbon and we're still accumulating carbon. Everything's kind of okay. Whereas in the mid-meadow, where the die-off was most pronounced, we do see that loss of carbon in the sediment and then the recovery with the recovery of seagrass shoot densities. We think this is because the seagrass in the northern part of the meadow are more resilient than in the center of the meadow, mostly due to their proximity to the inlet, which brings in cool oceanic water during high tide in the summer and provides this thermal stress relief to the seagrass, whereas in the center of the meadow, Oh, I lost it. No. It's continuously stressed. I have something to prove my point, if it works. Yes. So this is a temperature animation from 2 days in July 2017. And you can see the site near the inlet. When the tide comes in, it cools down. But in the center of the meadow here, it stays really, really hot. It stays also, in this case that I chose, above the thermal tolerance threshold for eelgrass, whereas in the northern part of the meadow, this doesn't happen for quite as long. So this is also important information for managers because the success of a seagrass restoration effort might be greater if it's in an area that has higher flushing and a shorter residence time so that you don't get that persisting thermal stress on the seagrasses. So this kind of wraps up that part of my talk that's about the carbon sequestration and storage in relation to this die-off event. Now what I want to do is just show a couple results from a paper of ours that got published yesterday. And that talks about how seagrass meadows are likely to respond to another part of climate change, and that's ocean acidification. So just quickly, the setup, there were some deployments done over multiple days in April and June 2015. And what you're looking at here is these data that were binned by hour of day to get a nice typical day-night cycle of our data for that period. And so you can see the diel variations in light driving the variations in benthic oxygen exchange, which in turn lead to almost perfectly out of phase oscillations in water column PCO2 and oxygen. And so what we're asking here is if photosynthesis is limited under conditions of high O2 and low PCO2, and inversely, if photosynthesis is stimulated under conditions of high PCO2 and low O2. The reason we're asking this is because there's a lot of discussion in the seagrass literature that suggests that under future scenarios of ocean acidification with increased CO2 emissions, that this might stimulate seagrass production and that seagrasses may in fact be winners in future ocean acidification scenarios. And we have some in situ data that can kind of chime in on that. So what we did to answer this question is we took the ratio of PCO2 to O2 and binned all the data according to this ratio into 4 groups, and it's their 25th percentiles of this ratio. And then we compared the data in the lower 25th percentile, that's all the blue bars, and the upper 25th percentile, that's the red bars. So on the very left, I lost it again, these are the ratios for those groups, and then we gave you the PCO2 values, the O2 concentrations as well, and then this is light for the 2 groups, and this is the benthic oxygen flux. And what we are highlighting here, first of all, there's no difference in light between the 2 groups, and that's very convenient. That means we can disregard the effects of light on flux, and allows us to see that these fluxes are not different between the 2 groups, which suggests that there's neither stimulation or limitation of seagrass photosynthesis based on the oscillations of O2 and PCO2. So that suggests that seagrasses may not, in fact, benefit from increased PCO2 levels. We did not see that in our study, at least. That was done under natural conditions. So just to conclude, we saw that restoration efforts reinstated carbon storage and sequestration in the meadow, but that carbon storage was very vulnerable to this die-off event as a result of temperature stress. The die-off event also caused a shift in trophic status from balanced to heterotrophic to autotrophic. We will see what happens in future years as things stabilize, hopefully, unless we get another heat wave. And then that there is some resilience due to the proximity to inlets and reduced thermal stress. And then finally, we didn't see any stimulation of photosynthesis at high CO2 concentrations. So that concludes my talk.
Great, that was a wonderful summary, and I'm sure there are lots of questions to follow.
Hi, I'm Katie Sparrow, Florida State. You said that the die-offs were related to mobilization of carbon that was thousands of years old. I was wondering if you could elaborate on that.
So, yeah, so this was like kind of an extrapolation for seagrass meadows globally. The carbon that we lost was accumulated over a period of about a decade. But there are other seagrass meadows, like in the Mediterranean, where they've been accumulating carbon over much longer timescales, up to millennia. And so my point was that, you know, with the potential loss of these ecosystems, we could be remineralizing carbon that has been stored for a very long time. So that's not the case in this type of, and not for Zostera beds, but for much slower growing species.
Other question?
Hi, Tricia Thibodeau, Virginia Institute of Marine Science. I was curious, since you looked at temperature stress and then you looked at CO2 stress, but in different sort of experiments, so to speak, like how you think they might influence each other, and if you think one might have an effect more immediately than the other.
Right, so so the CO2 part of this was done, I want to say almost like at a single point in time. Can I go back? I can go back. You see that temperature didn't vary that much over that period that the CO2 was measured. So I don't think we can really like relate the two stresses. One thing that I do have to say is that we didn't see any effect of CO2 at this time scale. That doesn't mean that with an increase in background levels that we wouldn't see an effect. So.
Hi, Will Barrelson, USC. Really interesting talk. One question, though, that the balance of autotrophy and heterotrophy isn't terribly consistent with the accumulation of organic matter over the recovery period, is it? Or am I missing something there?
What do you mean?
How do you get the organic matter recovered in the sediments unless you have net organic production exceeding respiration?
Yeah, so it's true that we don't see that, and it kind of depends on what time scale you look at it on. Well, no, it's not out yet. It's in one of my papers that's in review. But we looked at what the flux should be to account for this accumulation of organic matter, and it's actually per day. Over like January to maximum biomass in June, and it's actually very, very small compared to the overall fluxes that we get. So it doesn't show up.
So in other words, it's a small fraction of the swing between respiration and production.
Yeah. So hi, George Walbusser, OSU. Really nice talk. It's great to see the sort of sub-atmospheric CO2 levels. We see similar things in seagrass beds in Oregon. But I guess a little bit, I was following on Will's question. So is an inference from what you've shown that really the primary way that the seagrasses bury carbon is by particle trapping? And then as sort of a contentious sort of follow-on is, so do we just need to filter water better coming out of the rivers, and that would be a way to sequester, because there's so much interest in blue carbon, right? But it's what your data suggests is it's really the particle trapping more than the actual net metabolism of the seagrass itself. Is that, or am I misinterpreting?
Yeah, it sure, at least for this system, seems like most of the carbon sink capacity comes from carbon burial. But metabolism is important in that most of that carbon that is buried is that seagrass biomass. So that is important as well.
Yeah, so I guess that's where I'm confused, because if you're maybe in the panel we could talk about it, but if it's a balance in the autotrophy, heterotrophy, then it's hard to see where the biomass accumulation comes from.
Right, well, so that always confused me too when I was creating the carbon cycle diagram, but I think it's important to keep in mind it's not just the seagrass metabolism that we're talking about. It does incorporate everything else that's in the ecosystem, so there are a lot of other things respiring that.
Thanks for clarifying that.